All right, hi everybody. Welcome to our lecture on cell membranes, or cells and cell membrane function. And so what we'll look at today is we'll start off by looking at a couple of different cell types. There are two primary categories of cells on this wonderful planet. One category of cell is always single-celled, and they're small. They don't have a nucleus. They don't have compartmentalized reactions in their cell. They're just like little bacteria floating around. And so we call these prokaryotic cells. And so prokaryotic cells are small, they're tiny, they don't have nucleus or organelles. They're always single-celled. They're pretty primitive in function, although you would be surprised at some of the complexities that they have going on inside them. Some examples of prokaryotic cells are true bacteria or eubacteria. It's an entire domain of life. So consider that. Consider that animals, plants, fungi, and protists make up one domain of life, and there's an entirely separate domain of life that is made up of entirely bacteria. And so that's where we get like our bacteria, like staph infections, MRSA, that kind of stuff comes from the eubacteria. Also, like your gut biome, the bacteria that live in your gut and feed on some of the food that you eat, those are also part of the prokaryotic organisms and the eubacteria domain. There's another domain of life. These ones are quite interesting. They're actually more closely related to eukaryotic cells, or um, we should say eukaryotic cells are more closely related to these guys than they are to prokaryotic cells, even though these are prokaryotic cells. And so it's an entire domain called archaea, which is an extremophile. They love extreme environments, so high pH, low pH, high heat, things like that, high acidity, that sort of stuff. Okay, so the other type of cell, so we have one type of cell is prokaryotic. It's pretty simple, it's really small, it lacks a lot of structure, compartmentalized structures. The eukaryotic cells are much, much bigger. They're almost like on average, right? A eukaryotic cell is 10 times the size of a prokaryotic cell. They do have a nucleus, they do have organelles. Some are single-celled and some are multi-celled. So like when we talk about like protists are single-celled organisms that behave and kind of, I would just say, have a personality like some of the other multicellular kingdoms of life. So a domain is bigger than a kingdom. So the domain of eukaryota, the domain, incorporates multicellular plants, animals, and fungi, but it also has this little compartment, kingdom, if you will, of protists. And each protist kind of resembles one of those other multicellular kingdoms. So there's like animal-like protists, there's plant-like protists, there's fungi-like protists, and stuff like that. What's very important about eukaryotic cells is they do have a nucleus, so they enclose their chromosomes, their DNA, inside of another membrane inside of the cell membrane. So it's like you have to go through the cell membrane, then through the nuclear membrane, just to get to the nucleus, just to get to the DNA. They also have a bunch of organelles, so these are membrane-bound organelles. So they are also surrounded by a phospholipid bilayer. And most importantly, what this does for the eukaryotic cell is it allows it to become much more complex and advanced in its function because it can compartmentalize reactions. So it can take like one of these vessels, right, one of these organelles, and it can make the inside of it extremely acidic, so acidic that it breaks down anything that enters it. And so because it can compartmentalize these reactions, it allows for the cell to become much more sophisticated and complex. And so that's why we see most likely organisms that are multicellular, like us and plants and fungi and stuff. They are only that way because of the compartmentalization of the reactions that take place in their cells. So here's a little tree of life, if you will. It shows the three domains that we were talking about. The color of purple here on the left shows you the prokaryotic prokaryotic domain. And it's rooted down here. What you'll see is this black line represents the universal common ancestor of all living things, right? And so presumably it started quite simple, like prokaryotic life. Even though these are still around today, they were probably some of the first ones on the planet. And then there was a split and you get extremophiles. So that's all these like the Archaean domain, or the green. And the Archaea domain broke off from the eukaryotic domain after it had already split from the prokaryotic domain over here with the true bacteria. So archaea, the extremophiles, split off from the regular bacteria, and then from the extremophiles split off what is most familiar in life to you today. So that would be animals, plants, fungi, and protists. So let's take a look at these cells and kind of some of the components that they have inside of them. A prokaryotic cell is complex. It's just not nearly as complex as a eukaryotic cell. Now, the prokaryotic cell in this image has been expanded. It's been blown up to about 10 times as big as it should be if it was going to be proportional to the eukaryotic cell. So prokaryotic cell is much smaller. A lot of them have a flagellum, so that's like a whip-like tail that can help them move around. Some of them don't, you know. They've got a nucleoid, so it's kind of like a nucleus-like material. It's kind of like a central area where it keeps its chromosomes. Plasmids are the circular chromosomes that prokaryotic cells use. That's where they store their DNA. And then if we look over here, the eukaryotic cell is going to have a nucleus, and the nucleus is where it keeps its DNA. It's going to have an endoplasmic reticulum. So you got a rough endoplasmic reticulum, which has ribosomes studded all over the outside of it. It's got a Golgi apparatus. It's got smooth endoplasmic reticulum. Lysosomes help to break things down. Centrioles help for cell division. Mitochondria help for energy. So if you look at the eukaryotic cell, it's got all these compartmentalized organelles that each carry out a specific reaction. And that's what allows the eukaryotic cell to become so much more complex and regulated than a prokaryotic cell. Okay, so here's some of the membrane functions. So all cells are surrounded by a plasma membrane or a phospholipid bilayer. And those membranes can carry out several functions. So you can think about it sort of like a sieve. Cell membranes are semi-permeable, meaning that some stuff can get in easily, some stuff it requires a lot more effort to get it in. And same thing with leaving. So semi-permeable, some stuff comes in easily, some stuff comes in out with some effort. So here's some of the functions that they carry out. Transport, right? So you can analogize a cell to like a country or a city or a state or something like that, right? Some sovereign chunk of society, right? So that could be like a country. And so a country is going to have closed borders, just like a cell membrane, but a country's borders are going to be semi-permeable, meaning we allow some things to come in quite easily, but other things we highly regulate how much they come into our country. And so that's one of the things the cell membrane does is it highly regulates how things come into it and how things leave it. Also, inside of the membrane, there will probably be enzymes that either build things up or break things down, and that's embedded into the membrane itself. You've got signal transduction, so like if some signal comes from outside the cell and interacts with some of the surface membranes on the cell, that signal can be transduced. We talked about this in a previous lecture, but that signal can be transduced, meaning it's sort of like somebody's outside your house, they knock on your door in Morse code, you hear the Morse code, you write it down, you translate it, you send that message to somebody inside the house. The message didn't actually pass through the house. It was the signal was transduced through the walls of the house or the membrane of the house, if you will. So that's signal transduction. Cell-to-cell recognition. So anytime like your immune system is fighting off a virus, it's going to try to travel around and find cell membranes that are not recognized as self. So your immune system is really good at going around and having a secret handshake with all of the cells in your body. If it comes across one that messes up the handshake, then it releases some antibodies. This is kind of a simplification, but antibodies get released. Those antibodies stick to that weird cell, and that signals for another like giant monster cell to come and swallow it up and eat it. And so that's typically how we fight viruses. Viruses are covered in antibodies. Antibodies signal for this giant it's like a white blood cell basically comes in, swallows that thing whole, digests it up, and eats it. So cell-to-cell recognition is important, right? Recognizing self from others, especially with immune function and stuff like that. And then also intracellular joining. So like how your skin cells attach to each other would be like how the intracellular joining, and it would be done through the cell membrane. And then also attachment from the cytoskeleton, which is inside the cell, the rope-like structures that we talked about in the last unit, and then how those are connected through the membrane to the extracellular matrix, all the stuff outside the cell, all the structural elements out there. Okay, so we're going to watch a quick video on membrane transport.
Animal cells are enclosed in a plasma membrane, which consists of two layers of phospholipids. The hydrophobic nature of the cell membrane makes it intrinsically permeable to small non-polar and uncharged polar molecules, but non-permeable to large polar molecules and charged particles. Charged particles, such as ions, must use special channels to move through the membrane. Transport of a molecule can be passive or active. Passive transport does not require energy input because it moves the molecules downhill, for example, from higher to lower concentration. Active transport, on the other hand, moves the molecules against their gradients and therefore requires energy expenditure. Ion channels permit passive transport of ions. These are transmembrane proteins that form pores for ions to pass through. Most ion channels are specific for a certain type of ion. Ion channels can be classified by how they change their open, closed state in response to different factors of the environment. Common types of ion channels include leak channels. These channels are almost always open, allowing more or less steady flow of ions. Examples are potassium and sodium leak channels in neurons. Ligand-gated ion channels. These channels open upon binding of a ligand. They are most commonly found at synapses, where neurons communicate via chemical messages, or neurotransmitters. An example is the GABA receptor, a chloride channel located on postsynaptic neurons. It opens upon binding to GABA, a neurotransmitter released by the presynaptic neuron, and allows chloride ions to flow into the cell. Voltage-gated ion channels. These channels are regulated by membrane voltage. They open at some values of the membrane potential and close at others. These are the channels that underlie action potentials in neurons and cardiac muscles. Active transport of ions is carried out by ion transporters or ion pumps. These are transmembrane proteins that pump ions against their concentration gradient using cellular energy, such as ATP. Most notable example is the sodium-potassium pump, which maintains the resting potential in neurons by pumping 2 potassium in and 3 sodium out of the cell. Another type of ion transporters, known as secondary transporters, do not use ATP directly. Instead, they move one ion down its concentration gradient and use that energy to power the transport of a second ion. Symporters transport the two ions in the same direction, while antiporters pump the coupled molecule in the opposite direction.
Okay, and so that's just a video. You can go back and look at that on the slides and watch it again. But in essence, what it's saying is that membranes are semi-permeable. How molecules get in through the membrane really depends on the molecule itself. The one that we're going to focus on now is going to be transport. So enzymatic activity, signal transduction, these are just images from the list of terms that I gave you earlier. But we're going to look at transport. All right. So channel proteins. Channel proteins are a type of transmembrane protein. Trans means from one place to another, right? Transit, you're moving, right? Transfer, you're taking something, moving it somewhere. Transducing, right? Knocking on the door, the signal comes through. So transmembrane just means something coming through and then the protein. So it's a protein that sticks through a membrane. And because it sticks through the membrane, that means some of that protein is exposed to the outside of the cell and some of the protein is exposed to the inside of the cell. Now, some of these proteins can act like channels or tunnels, if you will, through the cell. And these channel proteins, some of them are called ion channels. And so that allows for polar ions, right? Because ions are charged particles. They're, like a sodium ion is a Na plus, so it's got one more proton than electron. And so it dissolves easily in water, but it doesn't move through a membrane very well on its own. And so we create these channels for it, these tunnels. So an ion channel allows for polar ions to flow through it. And then aquaporins are little channels that allow for water molecules to travel through the cell membrane. And aquaporins are basically what keep your cells hydrated, right? If your cells start to get dehydrated, meaning they have less water on the inside, then water will move through the membrane, through aquaporins, and rehydrate the inside of the cell. You also have carrier proteins, right? And so carrier proteins can help with moving a large molecule through the cell membrane. One type of carrier protein is called a co-transport protein. And so this would be where something like insulin comes along. Like you get a spike in blood sugar, you've got a lot of glucose molecules in your bloodstream. Your pancreas releases insulin. Insulin acts as sort of a gatekeeper to the cell membrane. It opens up the cell membrane, opens up glucose channels to allow for the glucose to diffuse into the cell, so the cell can take glucose out of the bloodstream, but also so the cell can get some energy from that. And then also active transport. So active transport is this idea that sometimes we need to move molecules into a place where they're already squished pretty tight, and they wouldn't do that normally, right? So this has to do a lot with diffusion. You'll get this with your diffusion lab. The idea is if you have molecules in an area of high concentration, right? So maybe you have like you put a bunch of air into a balloon, right? You blew air into a balloon. The air inside the balloon, the pressure is greater on the surface of the balloon on the inside than it is on the outside. And so if we were to release the mouth of the balloon, the air would flow spontaneously, requires no energy, would flow spontaneously from inside the balloon to outside the balloon. Well, the question is, how could you get more air in the balloon if it's already blown up? It would require energy to push air molecules into that balloon. And so that's the idea of active transport is anytime we have a concentration of molecules that's greater somewhere than it is somewhere else, and we're trying to move molecules into that area of greater concentration, kind of like blowing more air into a balloon, it's going to require energy from an outside source. And that energy source is typically adenosine triphosphate, or ATP. And then also we have voltage gated ion channels, and we find these a lot in neurons. You're not going to learn about them a lot in this class, but when you get into physiology, like animal physiology or human anatomy and physiology, you'll definitely go down the rabbit hole of neurons and how neurons reset. And they use voltage gated ion channels because your nervous system runs on electricity. So voltage gated ion channels is basically a regulated way of transducting a signal down a neuron, down a biological material, using ion channels. And the way that they reset the ion channel is just like a battery. You would take energy from ATP and use that to pump ions back across their concentration gradient to kind of like stacking them up on one side of the membrane or the other, kind of like blowing air into a balloon, right? And then it sits in that state of stasis until a signal comes along, a voltage change, and then that balloon gets released, and boom, that sends a signal down the neuron. But the most important part about all that, so you don't have to know anything about ion channels for this, or voltage gated ion channels, but they are reset. These voltage gated ion channels are reset by a specific type of transmembrane protein called the sodium potassium ion pump. And so the sodium potassium ion pump helps to reset the voltage gated ion channels. And this requires ATP, and it's called active transport. So here's a little image of a cell membrane, right? You got your phospholipid bilayer, and then inside that phospholipid bilayer, you have a transmembrane protein. Now notice this transmembrane protein has some primary and secondary structures to it. So there's your amino terminus, right? So the amino end of the protein molecule, and then you have your carboxylic end is over here. And so even though it's not pictured as beads on a string, this protein is just beads on a string that are interacting with each other that gives them folding conformational shape, three-dimensional structure that affects its overall functionality. And you'll see maybe there's a little bit of a beta sheet right here up here at the top. And then most of these are alpha helices, which are the secondary structures, right? And then the tertiary is how they all combine together to make the three-dimensional shape. Okay, we have aquaporins in action here in a little video. Somebody from the theoretical and computational biophysics group, yes, that's a real thing, right? Water molecules, right, these red and white spheres are trying to get through this transmembrane protein called an aquaporin. And so what these modelers done, these research modelers for biophysics, is they've said, okay, well, let's imagine body temperature. How much would these water molecules be moving around? And that's a Brownian motion. They're just bouncing around because they're at a certain temperature, right? And then they're trying to squeeze through this ion channel. And what you'll see is this protein for the aquaporin has been modeled by their biophysics team. And it's showing you how difficult, how challenging it is, right, for these water molecules to make it into the cell or make it out of the cell. And so there's like this fight going on in the middle of that channel um, for where they should go, like where the water molecules should go, right? And so that's how diffusion works. Is if you have a greater concentration of ions like salt inside of the cell, the water molecules outside the cell will fight their way into the cell until they can balance out the proportion of ions to water molecules inside and outside outside the cell until they reach equilibrium. All right, so active transport is fueled by the sodium potassium ion pump. And so what I have here is a little image or a little video of how the sodium potassium ion pump works. Oops.
Potassium pump is an active transport mechanism. Free sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and two of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, so let's take a look at what that looks like. So this is just an illustration of what that gentleman was talking about in the video. So you have the cytoplasm side, that's the inside of the cell, and you've got the outside of the cell. So you've got more sodium outside the cell than you do sodium inside the cell, and you've got more potassium inside the cell than you do outside the cell, or than you do sodium. Okay, well, that's true. You still have more potassium than inside than outside. So inside the cell, the sodium are going to find their way into this sodium potassium ion pump. They click in. As soon as they click in, ATP comes along and drops a phosphate group off on to the transmembrane protein, onto the sodium potassium ion pump. Now that phosphate group, if you remember, it's extremely electronegative. So it's got a lot, it's a negative ion, right? So it's got a lot of electron energy kind of pushing back on any other electrons in this whole structure. And that's what that's going to do, is change the way this structure is shaped. And it's going to bend and twist, and the sodium ions are then going to pop off and go outside the cell where the sodium ions were actually higher in concentration. Once the sodium ions are released, the potassium ions that are outside the cell in low concentration, right, come and bind with an affinity for this transmembrane protein. Then the phosphate group is going to pop off, which changes. Look, notice the cell looks like a Pac-Man facing up. When that phosphate group pops off, that negative energy leaves. Sounds like it's spiritual. The negative energy leaves and it changes its shape. So the negative energy leaves, it changes the protein shape, and then the Pac-Man faces down into the cell, and the potassium ions pop off. And so and then the whole cycle starts all over again. So that's the sodium potassium ion pump. Okay, so that pretty much wraps up our lecture on the cell membrane, osmosis, channel proteins, that sort of stuff. Yeah, look forward to having another lecture with you. This should be the last one for this unit as far as PowerPoints go. I am going to post another video where I draw a cell, and you're going to be asked to draw a cell as well. And then you'll post that in a discussion group. All right, see you guys later.